Imagine going to the doctor because there is like a raging fire in your gut, and the only solution they have is basically a blindfolded guessing game.
Right, which is a terrifying thought.
Exactly. But for millions of people out there suffering from inflammatory bowel disease, so conditions like Crohn's disease or ulcerative colitis, I mean, that is just their stark reality.
Yeah, the standard clinical approach is what we call a step-up treatment algorithm.
Right, so you start with the mild stuff, mild anti-inflammatory drugs. And if those fail, you step up to global immunosuppressants like steroids.
And if those fail, you bring out the heavy artillery, the targeted biologics.
Yeah. You try a drug, you wait for months, and you just sit there and see if the agonizing flare-ups stop. And the statistics are just brutal. Up to 30% of patients fail to respond to those first-line therapies right out of the gate.
Which is a huge number.
It is. And honestly, perhaps the most devastating part is that up to 50% of people who actually do find initial relief, they eventually lose that responsiveness over time. The fire just, it finds a way around the chemical roadblock.
Yeah, and that really points to a glaring gap in our biological understanding of IBD. I mean, the reason we see such high failure rates in the clinic is that historically we've only been studying the gut through static snapshots.
Like looking at a photograph instead of a movie.
Exactly. We examine a biopsy after the disease has already completely taken over the tissue. We see the charred remains of the forest, right? But we haven't had a reliable way to watch the initial spark actually catch, spread, and evolve in real time.
Until today.
Yes.
Because today we are looking at a map that finally takes the blindfold off. For this deep dive, we are exploring a massive, multi-resolution transcriptomic atlas. It was published in the journal Immunity in 2026 by Francerson and colleagues.
And this paper is just incredible because they didn't just take a single snapshot of the damage. They mapped the exact kinetics, the real-time movement and evolution of chronic colitis,
using some of the absolute most advanced genetic sequencing available today. So our mission for this deep dive is to take that dense stack of data and translate it into a clear map. We want to chart this chaotic landscape across both time and space.
Because we are going to uncover exactly how the physical structure of our intestines and our immune cells completely rewire themselves to sustain a chronic disease.
Okay, let's unpack this.
So to understand the physical space of the gut, we really first have to map the timeline. We need to know how chronic inflammation initiates and compounds over the span of weeks.
Right.
And to do that, the researchers utilized two distinct, well-established mouse models to recreate the layered complexity of human IBD. Because, human IBD is highly heterogeneous.
Meaning it doesn't always start the exact same way in every single patient. So just injecting a single chemical to make a mouse sick wouldn't actually reflect the reality of the disease in humans.
Not at all. It wouldn't be accurate. So the first model they used is called the T-cell transfer model. In this scenario, researchers take naive T cells.
And those are immune cells that haven't been programmed to attack a specific target yet, right?
Right, exactly. They're like blank slates. They take these naive T cells and introduce them into hosts that completely lack their own immune system.
Oh, wow. So there are no normal regulatory cells there to act as peacekeepers.
Bingo. Because those peacekeepers are missing, the introduced T cells just rapidly expand. They essentially look around the gut, they mistake the normal, everyday bacteria for a massive pathogenic threat, and they just launch an all-out adaptive immune attack.
So this model is essentially showing us how a highly specific immune system goes completely rogue.
Yeah, it simulates that active-aggressive attack.
But, IBD isn't always caused by a hyperactive immune system going on the offensive. Sometimes it's a failure of the body's natural brakes, right?
Which leads us directly to the second model they used, the spontaneous knockout model. These mice, they are genetically engineered to lack a very specific protein, a crucial anti-inflammatory chemical signal called IL-10. Normally, this IL-10 signal acts as like a biochemical megaphone constantly shouting, "Stand down, the environment is safe."
So without that megaphone, the mice just have a defective immune tolerance.
Exactly. They spontaneously develop colitis over time because they simply cannot stop reacting to standard environmental triggers.
And to ground all of this in actual human medicine, the researchers also introduced a clinical intervention, didn't they?
They did. They treated some of these mice with a targeted biologic antibody, anti-IL-12p40. This is the exact same kind of drug used to induce remission in actual human IBD patients.
Okay, so it intercepts a specific inflammatory pathway. It essentially blocks the chemical text messages the immune cells use to coordinate their attack.
Right. And this experimental design is brilliant because it allows us to track the baseline disease progression, and then observe how the tissue dynamically responds when a specific inflammatory pathway is completely blocked.
And to capture this overarching timeline, they began with something called bulk RNA sequencing.
Yes.
If we use a visual memory hook here for you listening, bulk RNA sequencing is like taking a satellite time-lapse of a massive forest fire over the course of several months. You're measuring a huge area of the gut all at once.
You can see the overall heat signature changing.
Yeah.
Where the fire is spreading, how intense the blaze is getting.
But you cannot see the individual trees burning or the individual firefighters on the ground. You are just capturing the total sum of all the genes being turned on or off in a large chunk of tissue.
And that satellite view revealed a fascinating, basically opposing dynamic. Over time, across both of these distinct mouse models, there was a progressive, massive downregulation of genes responsible for tissue remodeling and regeneration.
Wait, so the gut was fundamentally losing its structural ability to heal itself?
Yes. The healing pathways were shutting down, and simultaneously, the signals for inflammatory pathways were just skyrocketing.
It's like the biological construction crews were being laid off at the exact moment the arsonists were working overtime.
That's a perfect way to put it. But of course, mouse models are only useful if they actually reflect human suffering.
Right, they had to prove this wasn't just some weird quirk of rodent biology.
Exactly. So they took these distinct inflammatory gene signatures from the mice, and they cross-referenced them with actual biopsy data from human ulcerative colitis patients. They were actively looking for overlap.
And they found a specific set of conserved genes, right? 138 completely conserved genes, to be exact.
Yes, 138 genes that were significantly altered across all the mouse models and in the human patients.
Which is a huge deal. I mean, the concept of genetic conservation is basically the holy grail in this kind of research.
It really is. Because if a gene's behavior is identical in a mouse's gut fire and a human's gut fire, it means that gene is not just a random bystander. It is a foundational, non-negotiable pillar of the disease mechanism.
Evolutionarily, it is a core driver. And when they analyzed these 138 pillars, they found that they were heavily enriched for chemical alarm bells and the homing signals that call in specific shock troops of the immune system.
Specifically, neutrophil chemotaxes and various cytokines.
Okay, let's pause here for a quick recap to lock in what we've covered so far. If you're taking mental notes, here are the core takeaways from this timeline of chaos. Bullet point 1. Human IBD is a guessing game because we usually only see the aftermath of the inflammation.
Right, the static snapshots.
Bullet point 2. The researchers used two distinct models, one simulating an active immune attack and one simulating a loss of the body's natural anti-inflammatory brakes.
The T-cell transfer and the IL-10 knockout.
Bullet point 3. A satellite view of the tissue revealed that as inflammation spikes, the gut's ability to heal simultaneously shuts down. And bullet point 4. They identified 138 foundational genes shared between mice and humans that orchestrate this exact specific type of destruction.
Spot on.
So here's a question for you listening. If we know the forest fire is growing and we know the overarching alarms sounding off, is that enough to put the fire out?
Well, the clinical failure rates we talked about earlier tell us it's absolutely not. Knowing the fire exists and knowing the general alarms, that doesn't tell us who is setting the fires or who is failing to extinguish them.
Right.
We have to zoom in from that satellite view down to the street level. We need to analyze individual actors. And that requires single-cell RNA sequencing.
And this technique changes the game entirely. I mean, instead of grinding up a whole piece of tissue into a smoothie and looking at the average, you physically separate the tissue into over 600,000 individual cells.
Unbelievable resolution.
You read the RNA of each cell, one by one. It allows you to profile the exact identity, the job description, and the current mood of every single cell in the gut at different points in time.
The granularity really is staggering. And when they looked at the epithelial compartment, so the cells that physically line the gut, they uncovered a profound shift in cellular behavior.
What kind of shift?
Well, as the disease progressed over weeks, these barrier cells progressively activated genes dedicated to antigen presentation. Specifically MHC class II genes like H2AA and H2AB1.
Okay, let's break down why that is so shocking. Because epithelial cells, they're supposed to be the passive brick wall of the gut.
Right.
Their primary job is structural, secrete mucus, absorb nutrients, and just keep the trillions of bacteria on the inside of the digestive tube from leaking into the bloodstream.
Yeah, they are the bricks.
But antigen presentation, on the other hand, is the process of holding up little protein fragments, like biochemical wanted posters, to show the immune system what to attack. Usually, handing out those wanted posters is the exclusive job of specialized professional immune cells.
Exactly. Professional immune cells, like dendritic cells or macrophages, they patrol the tissue, looking for invaders to present. But this single-cell data proves that during chronic colitis, the epithelial cells themselves are actively participating in the immune response.
Wait, really? The physical wall of the gut?
Yeah, the literal physical wall. The bricks in the wall are suddenly sprouting arms and holding up their own wanted posters. They are directly, actively interacting with the immune system.
Wow. Which fundamentally changes the treatment paradigm, because if the structural cells of the gut are actively poking the bear, you can't just suppress the immune cells and expect a cure.
You can't, because the physical environment itself is driving a feedback loop that just amplifies the inflammation over and over.
It forces us to view the gut not as a victim of the immune system, but as an active co-conspirator.
It does. And speaking of the immune system, that single-cell street-level view also completely reshaped our understanding of the primary attackers.
Oh, the immune cells themselves.
Yeah. Alongside the memory and cytotoxic CD4 plus T cells that actually initiate the attack, the researchers documented a massive dynamic expansion of neutrophils.
Now, neutrophils have a bit of a reputation in biology. The classical view is that they are the blunt, disposable foot soldiers of the immune system.
Very blunt.
They rush to an infection, release a toxic payload of chemicals to just kill whatever is there, and then they die rapidly. They were basically biological cluster bombs.
Right. But the single-cell RNA sequencing revealed that in chronic colitis, neutrophils are anything but generic. They exist in highly heterogeneous, specialized states. They are literally forming specific tactical squads.
So they aren't all just detonating blindly.
Far from it. The researchers identified distinct subsets. One subset existed in a hyper-aggressive amplifier state. These neutrophils were marked by genes like CFCL3, and they were churning out specific proteins designed not just to kill bacteria, but to aggressively recruit even more immune cells to the area.
So they are actively feeding the cycle of inflammation.
Yes. And crucially, they identified another specialized subset of neutrophils that was entirely dependent on the specific chemical pathway targeted by the biologic drug we mentioned earlier.
The anti-IL-12P40 drug.
Exactly. This drug-dependent subset was marked by genes like WFDC17 and S100A8. And when the researchers treated the mice with the targeted biologic antibody, this specific population of aggressive neutrophils just plummeted.
Which beautifully explains the physical mechanism of the drug. The biologic isn't just generally calming the immune system down, it is selectively starving a very specific, dangerous squad of neutrophils of the chemical signals they need to survive.
Yes, it's precision starvation.
Okay, let's hit our second quick recap to lock in this street-level view. Here are the core bullet points. Bullet point 1. Single-cell sequencing let researchers look at 600,000 individual cells to see their exact behaviors.
Down to the individual cell.
Bullet point 2. The passive epithelial brick wall of the gut is actually a co-conspirator actively holding up wanted posters to fuel the immune attack.
The epithelial immune crosstalk.
Bullet point 3. Neutrophils are not just generic foot soldiers. They organize into highly specialized tactical squads. And bullet point 4. Some of these squads aggressively amplify inflammation, while others are the direct targets of our best clinical drugs.
Perfect summary.
So here's the next question for you to ponder. We now know when the inflammation happens, and we know exactly which cellular suspects are driving it. But the gut is essentially a 25-foot-long tube in humans.
It's massive.
Are these specialized hit squads just swimming around randomly, or are they setting up specific camps?
Well, to map the actual battlefield, the researchers deploy the final and, frankly, most groundbreaking technique in this entire study, spatial transcriptomics.
Which bridges the gap between molecular biology and physical anatomy.
Yes.
Think of it like adding GPS coordinates to our street-level view. The researchers used a spatial technique, the Visium technique, which is affectionately known in the lab as the Swiss roll.
I love that term. Because instead of taking a tiny localized cross-section of the gut, you take the entire length of the colon, roll it up onto itself like a pastry, and you slice it.
Right. And this method places a cross-section of the entire colon from the proximal end near the small intestine all the way down to the distal end near the rectum onto a single microscope slide.
It's incredible. And onto that slide, they apply specialized technology that contains thousands of microscopic barcoded spots.
And those barcodes act as GPS tags.
Exactly. When the RNA from the tissue sticks to the slide, it binds to a barcode.
Yeah.
So the researchers can sequence the RNA and map it back to exact physical X and Y coordinates on the slide.
So they can look at a high-resolution image of the gut and see precisely which genes are turned on in which physical locations at day 0, day 30, and day 50 of the disease.
And this spatial data completely dismantled a long-held assumption in the field of gastroenterology. Because previously, scientists assumed that these models induced a uniform pancolitis.
Meaning the entire gut was equally inflamed, like a uniform blanket of fire spread across the whole tissue.
Right. If the colon is a major city, the assumption was that the entire city was burning at the exact same temperature. But the spatial GPS data showed us that the destruction is actually highly localized.
Instead of a uniform fire, there are specific microclimates or neighborhoods of chaos. One city block is experiencing a catastrophic flood, another block is in the middle of a violent riot, and a third block is actively building makeshift military forts.
That's a great analogy. The spatial mapping allowed them to define 3 distinct inflammatory neighborhoods. The first was localized to the upper part of the colon, the proximal end. This neighborhood was characterized by an extreme epithelial antimicrobial response, marked by genes like DUOXA2 and RED3B. The barrier cells here were just churning out intense stress signals and proteins designed to carpet bomb bacteria.
So neighborhood number 1 is the proximal stress zone.
Exactly. Then, moving down the tissue, they found a completely different inflammatory signature dominating the distal colon, the lower part of the gut. The genetic profile of the destruction here was entirely distinct from the upper colon.
It's like a different city block with a different crisis.
Right. But the third neighborhood is perhaps the most critical for understanding why this disease is so chronic and stubborn. They discovered regions defined by tertiary lymphoid structures, or TLS.
Oh, here's where it gets really interesting. Because your immune system normally coordinates its attacks from secondary lymphoid organs, things like your lymph nodes or your spleen.
Those are the official permanent military bases of the body.
But a tertiary lymphoid structure is the immune system deciding to build an unauthorized makeshift forward operating base right inside the infected tissue itself.
Yes. It organizes the immune cells right on the front lines for a sustained, localized response. And under classical immunological theory, we believe these structures heavily depend on B cells to form the architectural foundation.
Like the B cells act like the combat engineers building the base.
Right. But due to the specific nature of the immunodeficient mouse models used, the researchers observed something astounding. These TLS-like structures were forming and creating severe inflammatory niches in the complete absence of B cells.
Wait, they are building the forward operating base without the engineers? How is the body pulling that off?
By utilizing alternative genetic blueprints. The spatial data showed that specific genes like CCL20 and CLU were creating local microenvironments, chemical anchors in the tissue, that physically drew in T cells and dendritic cells.
Wow.
The tissue itself was spatially organizing the inflammation, clustering the attackers together to sustain the damage without even needing the traditional cellular architecture.
If we connect this to the bigger picture, it explains so much about the human experience of IBD. The disease isn't just one monolithic thing happening everywhere at once.
No, not at all.
It is an evolving ecosystem of distinct inflammatory neighborhoods, driven by different specialized cells, using different genetic blueprints, clustered in different physical locations along the gut.
And as the spatial maps from day 30 to day 50 show, these neighborhoods constantly shift, they migrate, and they compound their damage over time.
It's just wild. So let's do our final synthesis. Time for recap number 3. We started this deep dive exploring the murky, frustrating diagnostic landscape of inflammatory bowel disease. We asked why current treatments, which rely on throwing broad systemic immunosuppressants at the entire body, fail so frequently.
And this multi-resolution atlas provides the definitive answer. The treatment fails because the disease isn't static. It is a highly dynamic spatiotemporal evolution.
You have structural barrier cells actually learning to hand out wanted posters. You have specialized squads of neutrophils that adapt and change their tactics week by week.
And they are operating in very specific, physically localized neighborhoods that require different interventions.
For anyone listening who is navigating Crohn's or colitis, this research represents a monumental shift toward genuine precision medicine.
It really does. Future therapies won't rely on blunt force. By utilizing spatial and single-cell blueprints like this, clinicians will eventually be able to target the exact right cell, like those hyperaggressive neutrophil squads, at the exact right time in the disease progression.
And in the exact right physical location in the gut, we can learn to intercept the disease before it builds those localized fortresses of inflammation.
It is a total paradigm shift in how we view the biology of the gut.
It is. And it leaves me with one final lingering thought for you to carry with you today. We just learned that the passive structural cells of our intestines, the epithelial barrier we thought was just a dumb brick wall, can actively learn to present antigens and act as the invisible architect of chronic, localized inflammation.
Right. The physical barrier itself becomes the instigator.
So if the wall of the gut is capable of orchestrating an autoimmune response, well, could other barriers in our body be doing the exact same thing in secret?
Oh, that's a chilling thought.
Think about it. Could the blood-brain barrier, or the structural cells of our skin, or the delicate lining of our lungs be acting as the hidden architects behind other mysterious, incredibly difficult to treat autoimmune diseases?
Are the physical walls of our bodies actively participating in illnesses we currently blame entirely on the immune system?
That is a profound biological question. And with spatial transcriptomics rolling out across laboratories worldwide, it is a question we might just have the answer to in the next decade.
I wouldn't be surprised at all.
Definitely something to mull over the next time you think about how the intricate machinery of your body is put together. Thank you for joining us on this deep dive into the spatiotemporal atlas of chronic colitis. Keep questioning the world around you, keep looking past the static snapshots, and we'll catch you on the next one.